Hello and welcome to this GeoMx Academy tutorial. In this session, we will introduce how to normalize GeoMx protein data. Before we get started, I would like to introduce you to the custom scripts feature in our software that we will use to generate the visualizations for this tutorial. The custom scripts feature enables the creation of figures within the DSP analysis suite to supplement your existing interactive visualizations. To load a custom script, we will start by selecting Custom Scripts, Manage, Add, the plus sign, and then we will choose our script of interest, followed by Save. At this point, the script has been loaded into our DSP analysis suite. We can simply give it a name, click Add Again, and now the script is available to us in our analysis suite. Now that the script is loaded, we can run it. Please note that the script will apply to whatever dataset is selected in your DSPDA. This particular script is intended only for the QC dataset. This is because we will be using these figures on pre-normalized data to ultimately determine what normalization strategy to select. To run our script, we will simply select Custom Scripts, select our Evaluate Normalization script, and then click Run. At this point, we can go to the Dataset Summary tab in our Visualization panel, select Attachments, and see our available outputs. With that, let's get started with this Introduction to Normalization tutorial. Normalization is an important step that enables you to account for different technical variables in your experiment. These include differences in ROI or AOI sizes, slide-to-slide antibody binding, and tissue quality, just to name a few. Thus, normalization ensures that you can subsequently focus on the real biological differences at hand, and is a prerequisite to performing downstream statistical analyses, such as clustering and differential expression. There are 4 methods for normalization, utilizing either housekeepers, background, area, or nuclei. Area or nuclei normalization, also known as area or nuclei scaling, are considerably less effective as normalization strategies and are not typically recommended. We will demonstrate why later in this tutorial. Housekeepers or background normalization, on the other hand, are the 2 primary techniques that can be used to normalize your protein data based on your given dataset. It is important to note that a given QC dataset should only receive one normalization. For example, we would not perform housekeeping normalization followed by background normalization. Such operations could either double normalize your data or annul the previous operation. So how do we select the normalization strategy for our GeoMx protein data? Let's answer this question with an example. Here we have a dataset with 2 colorectal cancer sections. We've selected multiple ROIs and segmented each ROI into tumor and stroma AOIs. To answer the question of normalization, we will look at a series of pairwise correlation plots generated from our QC data. As a reminder, this data has passed the QC step in our analysis suite. In other words, we will analyze the pre-normalized data that has been already assessed for N-counter readout quality and adjusted based on technical spike-in controls. By default, we would like to color our graphs by slide and AOI type. This lets us identify shifts in the plots and identify the sources of a weaker correlation or potential outliers. Additionally, we calculate the standard deviation of the log ratios between the 2 targets in a given comparison. This is our measure of concordance. The smaller the variance in the data, the stronger the concordance. The first plot we like to analyze is the pairwise correlation plot for our negative IgGs, or negative controls. These 3 off-target antibodies represent the background levels in our experiment. A given subfigure represents 2 targets plotted with respect to each other based on their location in the figure. Each data point represents the count value for a single AOI. Now looking at this graph, intuitively we expect an amount of dynamic range for these negative antibodies. This is a function of those technical factors we mentioned earlier, such as smaller and larger AOI areas and tissue differences. In this particular plot, we're looking to answer two questions. Are these negative antibody counts stable and high enough to be used for normalization? And two, are 2 to 3 of them correlated enough such that we consider background robust for normalization? At first, the concept of background counts needing to be high enough may seem counterintuitive, because we tend to think of a lower background as better for an experiment. However, for normalization, we need our counts high enough to have a degree of stability. An easy way to internalize this concept is to consider a low negative IgG count value for a given AOI. Here we see an AOI with a rabbit IgG and mouse IgG count of around 5. Due to even mild variability, this AOI could have very well had a count of 6. This small one-count deviation, however, could change the normalization factor and thus the endogenous protein count levels in this AOI after normalization by upwards of 20%, which is quite significant. Now, if we consider an AOI with double-digit counts, such as 50, it's easy to recognize that a small variability in background counts would not have a significant effect on the data after normalization. Thus, as a general principle, we tend to avoid background as a normalization strategy if counts for at least 2 negative IgGs are not above 20. Lastly, beyond background stability, we also look for at least 2 IgGs that are well correlated, such that we can take their geometric mean to represent background. In this particular study, it is clear that the background is below 20 for many AOIs, and additionally, likely as a consequence, these negative IgGs are less stable and lack a strong enough correlation between at least 2 of them to justify background as a normalization method for this data. Moving forward, we will now perform an analogous analysis for our housekeepers, S6, GAPDH, and histone H3. As a measure of on-target endogenous positive controls, housekeeper antibody counts are orders of magnitude higher than background, so we're not concerned with their stability. However, we will ask the question of whether 2 to 3 of these housekeepers are correlated enough such that we can use their geometric mean to represent our normalization factor. In this study, we observe that S6 and GAPDH are well correlated with each other, while histone H3 does not behave as well compared to its 2 counterparts. It appears that histone H3 is not as concordant as a function of the differing AOI types, tumor and stroma. Thus, we conclude that we will only use S6 and GAPDH as the housekeepers for normalization in this study. As a final view of the data to evaluate normalization, we will study the relationships between our different normalization options. Intuitively, the different normalization factors will be generally concordant. This can be easily understood by taking the example of differing AOI sizes. A larger AOI will naturally have more area for both housekeeper and background antibodies to bind, and thus, background and housekeepers will generally be correlated with each other and also with area. This plot also highlights the inherent drawbacks to an area or nuclei normalization approach. This can be understood by looking at 2 AOIs with similar areas. It is clear that this AOI has 4 times as much background as this AOI from a different tissue. We can illustrate a similar concept with housekeepers. This observation is an indication that the 2 AOIs are potentially binding to antibodies to varying degrees. Area normalization would consider these AOIs the same, given that they have the same area, and give them the identical normalization factor, while background or housekeeper normalization would account for these differences, because the normalization factor for the particular AOI would actually be based on antibody signal for that AOI. In conclusion, given the low background counts and concordance between the negative IgGs, and in contrast, the strong concordance between S6 and GAPDH, our housekeepers, we prefer a housekeeping normalization for this particular dataset. Looking beyond this study, I hope you have understood the general principles of how to evaluate what normalization strategy to select and apply for your GeoMx protein data. Based on your given dataset, housekeeper or background tend to be the preferred normalization options. In some instances, we observe that one method is superior to another, as we saw in this tutorial. In other scenarios, however, we find that either housekeeper or background normalization methods are attractive, in which case we encourage that you pursue the different options in parallel for comparison. To determine specifically which antibodies to use for a given strategy, please study the pairwise plots presented with your own data. Thank you so much for watching this GeoMx Academy tutorial.